Get this. You started as just one single cell.
Really?
Yeah, just one. And from that, you grew into the trillions of cells person you are today.
Yeah.
It's mind-blowing. And today, we are diving into the world of cell growth and division.
Right.
We've got some excerpts from a biology textbook to guide us on this journey.
Cool.
So how do we go from one to trillions?
Well, it's all orchestrated by these two processes, mitosis and meiosis.
Mitosis and meiosis.
Yeah, they're like the choreographers of life. They make sure that cells divide and replicate with incredible accuracy.
So there's a whole dance routine happening inside us.
Yeah, I mean, maybe not a dance routine,
Uh-huh.
but definitely a specific sequence of steps, right?
Okay.
So that's where the cell cycle comes in?
The cell cycle.
Yes, it's the master plan
Right.
that guides each cell through its life, from birth to splitting into new cells.
Okay, so let's break down this cell cycle.
Okay.
What are the major steps in this microscopic dance-off?
Okay, so imagine a cell's whole lifespan is like a circle. Over 95% of that circle is interphase.
95%.
Yeah, that's the prep phase,
Okay.
where the cell grows, it does its normal job, and it copies its DNA, like getting ready for the split.
So it's like the warm-up before the main event.
Yeah, exactly. Got to make sure everything's in order.
Right.
And interphase itself is split into 3 phases.
Okay.
First is G1,
You won.
or gap 1, where the cell just focuses on growth and its regular metabolic activities. Then comes the S phase. That's where the DNA replication happens. It doubles the cell's DNA.
Okay.
And finally, G2 or gap 2.
Gotcha.
Final growth spurt, protein synthesis.
Okay.
It's like a last-minute check before the performance.
So the cell's really busy during interphase,
Mm-hmm.
ticking off a to-do list. But what about the other part of this circle?
Yeah.
The M phase.
Sem phase.
That's where the actual division happens, right?
That is where mitosis takes over.
Mitosis.
Yeah, it's the process of creating 2 identical daughter cells from that original parent cell.
Okay.
Think of it like a cell cloning itself.
Oh, okay. Making sure both new cells get a full set of chromosomes.
So it's like the cell has a built-in photocopier for its DNA.
Pretty much, yeah. It's incredibly efficient.
Yeah.
And mitosis has its own 4-step routine.
4 steps.
Prophase kicks things off.
Okay.
Chromosomes condense, becoming these visible structures,
Yeah.
kind of like packing their bags for the move.
Uh-huh.
At the same time, these things called centrosomes, they help organize the division. They move to opposite sides of the cell.
Setting the stage for the big separation.
Exactly.
Okay.
Then comes metaphase.
Metaphase.
This is like the most visually stunning part.
Really?
All the chromosomes line up perfectly at the cell's equator,
Wow.
attached to these spindle fibers
Yeah, okay.
radiating from those centrosomes.
Okay.
It's like a perfectly choreographed lineup.
So precision is key.
Yeah, make sure each daughter cell gets an equal share of that genetic material.
Wow. And then what? Do the spindle fibers yank them apart?
You got it. Really?
That's anaphase.
Anaphase.
Yeah, the centromeres, the point where the chromosomes are attached, they split, and those copies are pulled to opposite ends of the cell.
So the chromosomes have split.
Yes.
They've gone to their designated corners.
Yeah.
What's the final act in this?
Okay, so telophase brings down the curtain.
Telophase.
The chromosomes relax back into their less compact form.
Okay.
New nuclear envelopes form around each set, and the nucleolus reappears. It's like the cells are unpacking and settling into their new homes.
So 2 new nuclei,
Yes,
each with a complete set of chromosomes. Does that mean we're done?
almost.
Okay.
We've got the grand finale, cytokinesis,
Cytokinesis.
dividing the cytoplasm.
Okay.
Animal cells do this by pinching in the cell membrane and eventually just split the cell into plant cells because they've got those rigid walls.
Yeah.
They form a cell plate that grows outward from the center to divide the cell.
That's wild. The whole process is so intricate.
Right.
It makes you appreciate the complexity happening inside us all the time. But why is mitosis so important?
Okay, so mitosis is essential for growth, repair. Think about healing a cut and replacing old cells.
Okay.
Every day, millions of your cells die naturally.
Oh.
And mitosis is how you replenish them. If it goes wrong, cells can start dividing uncontrollably,
Okay.
which is what happens in cancer.
That puts it into perspective. It's not just a cool process. It's crucial for our survival.
Yes, it is.
But what about meiosis?
Meiosis.
You mentioned it earlier.
Yes.
How does that differ?
So, meiosis is the driving force behind sexual reproduction.
Okay.
Unlike mitosis, which makes identical copies, meiosis generates gametes.
Gametes.
Yes, sperm and egg cells,
Okay.
and each with half the number of chromosomes as the parent cell.
Hold on, half.
Yes.
Why is that important?
Well, think about it. When gametes fuse during fertilization, The resulting zygote needs a full set of chromosomes,
Right.
half from each parent. So if meiosis didn't happen, they would just double with every generation, and that would be total chaos.
That makes sense. So how does meiosis do that?
So meiosis involves two divisions,
Cute.
meiosis I and II, but with only one round of DNA replication.
Okay.
And the steps, especially meiosis I's, are quite different.
Different how?
Okay, so meiosis I is where the magic of genetic diversity happens.
Ooh, interesting.
Instead of chromosomes lining up individually like in mitosis, in meiosis I, homologous chromosomes pair up,
Okay.
forming what are called tetrads.
Tetrads.
Yes.
So the chromosomes are getting together in pairs.
Yes, they are.
What happens next?
These tetrads exchange genetic material,
Okay.
like trading cards.
Really?
It's called crossing over.
Crossing over.
It shuffles the genetic deck, creating new combinations of genes.
Okay.
That's why you and your siblings aren't identical clones.
That's incredible.
Yeah, it's not just about dividing chromosomes, it's mixing them up too.
Meiosis is way more complex than I initially thought.
It is, right? This crossing over introduces like a mind-boggling amount of potential variation. So in meiosis I, we've got prophase I, which is actually divided into 5 substages.
5.
Yes, leptotene, zygotene, pachytene, diploteen, and dikinesis.
All right.
In leptotene, the chromosomes start condensing and become visible.
Okay.
Then during zygotene, the homologous chromosomes pair up to form those tetrads we talked about.
They're getting ready for that genetic swap meet.
Exactly, yeah. And then in pachytene, that's when the actual crossing over takes place.
They're really swapping segments of their DNA.
Yes, creating new combinations of genes on each chromosome, increasing genetic diversity. Then comes diploteen.
Diplocene.
The homologous chromosomes begin to separate but stay connected at points called chiasmata.
Okay.
Those chiasmata are like little markers where the crossing over happened.
So they're holding hands where they traded genes.
Yes, and finally in dikinesis, the chromosomes condense further, the nucleolus disappears, and the nuclear envelope breaks down.
Okay.
The stage is set for the first meiotic division.
The chromosomes are all dressed up for the big dance.
So what about metaphase tail? Do they line up in the middle, like in mitosis?
Do they?
They do, but instead of individual chromosomes lining up,
Right.
it's the tetrads that align at the equator.
So it's more like a couple's dance than a solo performance.
Perfect analogy.
Yeah.
Then comes anaphase, where those homologous chromosomes are pulled apart to opposite poles of the cell.
So each pole gets a full set of chromosomes.
Yes.
But they haven't split into individual chromatids yet.
Got it.
Right.
In telophase I, the chromosomes arrive at their designated poles.
Right.
The nuclear envelope might reform,
Okay.
cytokinesis might happen, leaving us with 2 daughter cells, each with half the number of chromosomes as the parent cell.
Okay.
But each chromosome still consists of 2 sister chromatids.
So we finished the first act of meiosis,
Yes.
have the chromosome number. What happens in meiosis 2?
So meiosis Tay-Sachs is similar to mitosis,
Okay.
but there's no DNA replication beforehand. So those sister chromatids making up each chromosome are separated, resulting in 4 daughter cells, each with half the number of chromosomes as the original cell.
So it's like mitosis on a smaller scale in meiosis 2.
Yeah, you could say that.
Okay, can we walk through those stages just to make sure I got it?
Absolutely. Okay, so prophase 2 is similar to prophase in mitosis. Chromosomes condense and the nuclear envelope breaks down.
Okay.
Metaphase 2, the chromosomes line up individually at the equator, just like in mitosis.
Gotcha.
Anaphase 7 sees those sister chromatids pulled apart to opposite poles.
Right.
And telophase 2 wraps things up with the chromosomes decondensing and new nuclear envelopes forming.
Okay.
Finally, cytokinesis happens, resulting in those 4 haploid daughter cells.
Wow, that was a lot.
Right.
Both mitosis and meiosis are like meticulously choreographed dances, each with its own unique steps and significance.
You said it.
Mitosis is crucial for growth, repair, and asexual reproduction,
Yep.
ensuring that each daughter cell gets an identical set of chromosomes. Meanwhile, meiosis is the engine of sexual reproduction,
Yes.
making sure the offspring inherit the correct number of chromosomes by having the number in the gametes.
Yeah.
And let's not forget the amazing genetic diversity created by that crossing over during meiosis.
Absolutely.
It's like nature's way of shuffling the genetic deck
Yeah.
and ensuring that offspring are unique.
Exactly. Both processes are absolutely essential for the continuation of life.
Right.
They ensure the faithful transfer of genetic information and generate the variation that allows species to adapt and evolve.
I'm really starting to appreciate the beauty of cell division.
Yeah.
It's a lot more complex and fascinating than I initially realized.
Uh-huh.
Our textbook also mentions that variations are important for evolution.
Right.
How do you think the processes of mitosis and meiosis contribute to variation within a species?
That's a great question, and it leads us to the amazing world of genetic variation. While mitosis faithfully replicates genetic material, it's meiosis, with its crossing over and random assortment of chromosomes, that introduces most of the variation we see.
So mitosis is like the copy machine,
Yes.
making sure the blueprints are preserved, while meiosis is the remix artist, creating new variations on a theme.
Precisely, and it's this variation that natural selection acts on,
Right.
leading to new traits and eventually new species.
Wow, that's amazing.
Yeah,
That's a deep dive for another time.
absolutely.
You've definitely left me wanting to explore that further,
Yeah.
maybe in a future episode. But for now, I think we've covered a ton in this dive into the world of cell growth and division.
We have.
It's been great.
Yeah, it has.
I've learned a lot.
Good.
So thanks for joining me on this deep dive into cell growth and division.
Thank you for having me.
It's been a pleasure.
Yeah, it has. It really is amazing when you think about it.
What's that?
We all started from just one cell,
Oh, yeah.
a zygote. When a sperm and egg cell fuse,
Right.
all the instructions to build a human being
Yeah.
in something microscopic.
It's mind-boggling. And then that one cell keeps dividing, making more and more cells,
Uh-huh.
all working together to become tissues, organs, and eventually a whole person.
Yeah, it really is like a perfectly choreographed ballet,
It is.
with each cell playing its part.
Makes you wonder, what determines how fast a cell divides? Do all cells divide at the same rate? Like, are my skin cells dividing as fast as the cells in my brain?
That's a great question.
Thanks.
The speed of cell division really depends on the type of cell and the organism.
Okay.
So, for instance, cells lining your stomach and intestines, they're constantly being shed.
Oh, wow.
They have to divide quickly to replace those lost cells.
Yeah.
But then, on the other hand, your nerve cells in your brain, they rarely divide after you reach adulthood.
So different cells have different internal clocks telling them when to divide.
Yeah, you could say that.
What happens to the cells that don't divide? Do they just hang out?
Well, some cells go into a state of quiet rest. It's called the G0 phase.
G0 phase.
Yeah, they're still working, still metabolically active, but they're not preparing to divide.
Okay.
Like your heart muscle cells, for example, they spend most of their lives in G0.
So they're just taking the breaks in the dance floor.
Yeah, but they're still part of the show.
It seems like there's so much variation,
Yeah, there is.
even in how cells divide.
And get this.
Right.
Some cells can even jump back into the cell cycle from GRO
Great.
if they need to, like when they need to repair damaged tissue.
That's cool.
Yeah, it's a fantastic example of how adaptable and flexible cells can be.
It all seems very organized and controlled.
Yeah.
Our textbook mentions that cell division is under genetic control.
Uh-huh.
What does that actually mean? Is there like a tiny instruction manual?
That's a key point, yeah.
Okay.
The whole process, from DNA replication timing to how the chromosomes are separated,
Right.
it's all regulated by this complex network of genes and proteins.
So there's like a control panel inside dictating when and how the cell cycle happens.
Pretty much, yeah. And this control system is vital for preventing errors
Right.
that could lead to cell death or
Yeah.
uncontrolled cell division, which is a hallmark of cancer.
Without these checks and balances, things could go haywire.
Yeah, definitely.
But what about things outside the cell?
Yeah.
Can things like what we eat or being exposed to toxins
Yeah,
affect the cell cycle?
absolutely.
Really?
External factors can definitely influence the cell cycle. Think about it. Certain nutrients are essential for DNA replication and cell division.
Okay.
So if you don't get enough of those, it can slow down or even stop the cell cycle.
Oh, wow.
So it's like the cell cycle needs the right fuel and maintenance to run smoothly.
Right. And what about toxins?
Exactly. Exposure to toxins, radiation, or even some viruses can damage DNA, messing up the cell cycle,
Okay.
potentially leading to mutations or cell death.
So it's like a delicate balance between the internal genetic control and the external environment. Our cells are constantly facing challenges.
They are, but they manage to keep things running smoothly most of the time.
Yeah.
Remember how you mentioned that you were fascinated by how plant cells could be both haploid and diploid?
Oh, yeah, that's right.
Well, in some organisms, like plants, both haploid and diploid cells can undergo mitosis.
That's right.
It's fascinating how different plants are from animals.
Yeah.
What's the significance of that? Well, remember, haploid cells have a single set of chromosomes,
Right.
while diploid cells have two sets.
Yeah.
Well, in plants, some stages of their life cycle involve haploid individuals called gainophytes,
Okay.
which produce gametes through mitosis.
So it's like plants have two different generations,
Mm-hmm.
one haploid and one diploid, both capable of undergoing mitosis.
Yeah, it's so different from animals,
It is.
where mitosis usually only happens in diploid cells.
It's a striking difference.
It is.
It highlights just how diverse life cycles and reproductive strategies could be.
Absolutely.
We've covered a lot of the mechanics of the cell cycle on
Mhm.
mitosis and meiosis.
Yeah.
But I want to circle back to something we talked about earlier, the importance of cell division for growth and repair.
Right.
How do those processes actually contribute to the development of a complex organism?
Think of it this way. It all boils down to the remarkable ability of cells to replicate themselves
Okay.
and then specialize into different cell types.
Uh,
Starting from that single fertilized egg, repeated rounds of cell division produce trillions of cells,
right.
each with the same genetic blueprint,
Yeah.
but with the potential to take on diverse roles.
It's like having a master builder, starting with a single blueprint and then creating a complex structure
Yeah,
with specialized rooms and functions.
precisely. And this specialization is guided by intricate signaling pathways
Okay.
and genetic programs that tell those cells to become specific types. Nerve cells, muscle cells, skin cells, you name it.
So it's not just about making more cells.
Right.
It's about making the right kinds of cells in the right places.
Yeah, it's a carefully orchestrated symphony of growth and differentiation.
But what about the exceptions? You mentioned earlier that some cells, like heart muscle cells, they rarely divide once we're adults.
Yeah,
Does that mean our hearts can't repair themselves?
that's a crucial point.
Okay.
It highlights the limitations of cell division in certain tissues.
Right.
While many tissues are great at regenerating, some, like heart muscle, have limited regenerative potential.
So if heart muscle cells get damaged, they can't just be replaced by new cells.
And unfortunately, that's often the case.
Mm.
When heart muscle is damaged, it often leads to scarring,
Okay.
as the body tries to repair the damage with fibrous tissue instead of new heart muscle cells.
That's kind of scary.
Yeah, so even though cell division is essential for growth and repair, it's not a cure-all. There are limits to what it can do. Yeah.
Understanding those limits is key for developing new treatments for diseases and injuries.
This is where stem cell research comes in, right?
Absolutely. Scientists are working on ways to harness the power of cell division to repair damaged tissues and organs,
Wow.
which could revolutionize how we treat a wide range of conditions.
That's like we're on the verge of unlocking the full potential of cell division, not just for maintaining our bodies, but for reversing damage and extending lifespan.
It's a fascinating field with so much potential.
Is
We've covered a lot in this deep dive, haven't we?
we have.
From the intricacies of the cell cycle, the differences between mitosis and meiosis,
Yeah.
the significance of cell division for growth, repair, and the development of complex organisms.
It's been quite a journey from a single cell to trillions,
Inhale it.
uncovering the very processes that make life possible.
Yeah.
And it's clear that the story of cell division is far from over.
As scientists continue to unlock its mysteries, we can expect even more groundbreaking discoveries that will change how we understand biology
Absolutely.
and open up new frontiers in medicine and biotechnology.
Indeed, we're at the forefront of some truly amazing discoveries.
That's great.
And on that note, we've come to the end of part 2 of our deep dive.
It's really remarkable how much we've learned
It is.
about the intricacies of life at the cellular level.
Uh-huh.
And yet, as with, well, practically everything in science,
Yeah,
there's always more to discover.
as always.
Our textbook really focused on
Yeah,
how mitosis and meiosis work,
right.
but it didn't go into what happens when those processes go wrong.
Oh, yeah.
So what can go wrong during cell division?
Well, while mitosis and meiosis are usually incredibly precise, errors can happen, and the consequences can be pretty significant.
Oh, no.
One possibility is that the chromosomes might not separate properly during mitosis or meiosis.
During either one.
Yeah, it's called non-disjunction.
Non-disjunction.
And it can lead to daughter cells having an abnormal number of chromosomes.
So what happens to those cells with the wrong number of chromosomes?
Well, it depends on which chromosomes are involved
Okay,
and how severe the imbalance is.
right.
Some cells with an abnormal number of chromosomes, they just might not be viable, they'll die off.
Okay.
Others might survive, but not function correctly, potentially leading to developmental disorders or even cancer.
So it's like a delicate balancing act.
Uh-huh.
Too few or too many chromosomes and it throws off the whole system.
Yeah, and there are other things that can go wrong during cell division.
Like what?
For example, DNA can get damaged during replication,
Oh,
which can lead to mutations.
okay.
Or the checkpoints in the cell cycle, they can malfunction,
Right.
allowing a damaged cell to divide.
It's like a complex machine with safety features,
Yeah,
but sometimes those safety features fail,
yeah,
and that's when problems happen.
that's a great analogy. And we can't forget about the influence of the environment.
Right, of course.
Exposure to radiation, certain chemicals, even some viruses, they can damage DNA,
Right.
disrupt the cell cycle,
Okay.
increasing the chances of errors during division.
So it's this constant back and forth
Uh-huh.
between the cell's internal machinery trying to keep everything running smoothly
Right.
and the external environment throwing wrenches into the works.
Exactly. This interplay between genes and the environment is a major theme in biology.
It is.
It highlights the complexity of life
Yeah.
and the challenges organisms face in maintaining that delicate balance.
It's amazing that our bodies can function at all with all these potential pitfalls.
It's pretty incredible, isn't it?
It is.
You know, our textbook also mentions that variations are important for evolution.
Yeah.
I'm curious, how do you think errors in cell division contribute to that variation we see within a species?
That's a great question. It seems counterintuitive that errors could lead to something positive.
Well, remember those mutations we talked about?
Yeah.
While most mutations are either harmful or have no noticeable effect,
Okay.
some can actually be beneficial.
Really?
Yeah, they might provide an advantage in a particular environment.
So it's kind of like a genetic lottery.
Uh-huh.
Most of the time, you don't win anything,
Right?
but every once in a while, you hit the jackpot.
Exactly. And those beneficial mutations are what fuel evolution.
Oh, wow.
They provide the raw material for natural selection to work on. Over time, these mutations can accumulate, leading to new traits and eventually new species.
That's fascinating. So even though errors in cell division can be detrimental,
Yeah,
they're also a source of innovation and diversity.
they are.
In the living world, it's like a double-edged sword.
It really is, and that's part of what makes biology so captivating.
Yeah.
It's this story of constant change, adaptation, the interplay between order and chaos.
Well, this deep dive has definitely given me a new appreciation for the complexity of life, even at the cellular level.
Absolutely.
You know, we've explored the steps of mitosis and meiosis, talked about why those processes are so important for growth, repair, and reproduction,
Yeah.
and even discussed the consequences of errors and how variation plays a role in evolution.
We've covered a lot of ground today, that's for sure.
We've journeyed from a single cell to trillions, uncovering the fundamental processes that make life possible.
That's right.
And as we continue to explore and learn more about cell division, we're bound to make even more exciting discoveries
Absolutely.
that will change our understanding of biology and open up new possibilities in medicine and biotechnology.
I agree.
Well, it's been fascinating exploring the intricacies of cell growth and division with you.
It has. It's been a pleasure.
And thank you all for listening to this deep dive into the microscopic world that makes life possible.
Thanks for having me.